Let me know when you can see it. Okay. So yeah, who is this Brian guy? So I figured in terms of like networking and stuff, these are all the things you can bring up with me. I am a Scotty, so I grew up in Wisconsin. I have had my own company, Learn Think Code, so that space. I've been at a startup with Carrot Health in terms of I was director of data engineering. But most of my time has been since 2011 at no at Novartis. So I'm a Novartian if you are as well. Um, Let me know. And because of that, I was able to move to Switzerland for 3 years. So if any expats want to commiserate. But currently I live in Minnesota, and the tech community here is mini star. The half of the world's medical devices are made in Minnesota. And so we have a community around med tech, biotech, life sciences called the Medical Alley. And another community I'm involved in is mini analytics, which is sort of like data science in the healthcare space that's here. So we have lots of communities. Additionally, I'm part of the Open Bioinformatics Foundation, OBF, the GA for GH. I mostly program in R nowadays. And because I work at a pharma company on the translational side, involved in the Pharmaverse, which is submitting to health authorities using R instead of SAS. And I'm the Twin Cities city captain for Bits and Bio. What I geek out about is acceptance test driven development. So kind of quality validation of software and data. Big data bioinformatics. So how do we do stuff at scale? Biomarkers, which we'll talk about, and just molecular biology in general. So for me, I wanted to talk about biomarkers. And really the reason why is that we're in the golden age of precision medicine. And precision medicine for me is the use of biomarkers, kind of omics, imaging, digital, exposure data, potentially social and behavioral data, all in the diagnosis, prevention, and treatment of disease. In terms of where do drugs come from, most people, I think, are familiar with this, but there's this notion of discovery, kind of that early data, and then getting approval to go into a human. And then you get your phase one, phase two, phase three trials. Then you can submit for approval. And there may be after approval studies, but also real world data, data mining, again, to kind of feed back questions. We call it back translation, back into research. And really for us, the question comes down to responders versus non-responders. On the research side, you are setting up experiments. And as Becky said, it's all about going, the simple little workflow that gets you to results. And those results tell you what experiments, what samples are responding or not responding. And typically that data looks like matrices of data. This is Bioconductor's summarized experiment. Data about your samples, data about your features, data about the experiment. But a lot of times that data ties back to a knowledge graph of protein-protein interaction or which proteins are tied to which transcripts, et cetera, et cetera. On the clinical trial side, and for research, many times your samples are similar, and you're looking for what changes, what protocol will give you the results you need. For a clinical trial, you've locked down your protocol, and now the humans are different. And so as you're doing this, you may collect different samples from participants on the trial at different time points. And you're also monitoring their progression, disease progression, et cetera. There's trial endpoints that you're looking at. The data that you're going to produce in the trial is defined by CDISC, and it is fairly structured. And any samples that you take will be recorded in the BE domain. It's one of the tables. But the output of running those through some process, some analysis, those files may not get sent into the health authority. but they could be used. They may be sitting at your CRO, or if you have it internally, you can use that for additional research. And that helps us figure out responders versus non-responders, or where does drug resistance maybe pick up in the course of the trial. In terms of real world data, now you've got the whole population. And are there biomarkers that could help with diagnosis or prevention? Then you've got treatments. And here biomarkers could help with disease subtyping or treatment protocols. Like second line, if your CD19 is over 50% of your cells are expressing that, then we're going to do this treatment. But in the end, the data that we've got is claims or EMR data, and there is some standards kind of forming around that. And then whatever other data you can get, and trying to pick out of that, there may be some biomarker data, but trying to pick out of that, again, features that differentiate responders from non-responders. So the challenge on the research side is most people just focus on the experiment. And a lot of the data is tied up in registration systems. That knowledge base is part of the registration system. But different places have different ontologies, and it's really difficult to combine data, which is why people talk about fair data. So at least it's documented. That doesn't make it any easier to combine. For clinical trial data, literally the structure of all the files, the STTM tables, domains, or the ATOM domains, their standards aren't really standards. They're more conventions and guidelines. And there are multiple stakeholders. There's therapeutic areas, there are health authorities, there are implementation guides that kind of help you decide how you should do it. But really every trial can be different. And then just in general terms, like as a data architect, it's not the greatest data model. So if you are familiar with CDISC, in the end, like the files get sent to the health authority as SAS V5 transport files, which is a proprietary format, but it also is like 8-character columns, 40-character labels. Like it's bad. It's like old data. It's being worked on, but that's a huge kind of a challenge. And then again, that additional research, that biomarker data may not be part of what gets sent into the health authority, and it just literally may be locked away. In terms of the real world data challenges, the claims and EMR is really not necessarily enough. And so then you may look at death data or cause of death data, and this is really hard to get. And like companies like Tempus, they will charge you more data, charge you more for it, because the state you die in owns your death data, and they charge for it. And so that charge gets passed on. But you can also look at health risk and care quality data, social determinants of health. Does somebody have transportation needs? Do they live in a food island? Things like that. You can combine it with census data, exposure data, air quality data, places of interest. Do they groceries, fitness centers, healthcare facilities, demographic data? Are they married? Do they have, own a dog? Surprisingly, that has a huge effect on your health. And patient reported data as well. But the big challenge is lack of biomarkers. So we're starting to see these national biobanks. So they started with the UK biobank, but you also have FinGen in Finland, all of us in the US, and then the Eden Darwin projects in Europe. They are trying to get all of their EMR data into OMOP format, and then adding germline variant data. So huge variant databases. 23andMe, Tempus, Flatiron, they're adding omics data to patient or with 23andMe, the surveys, but also with EMR and EHR data. There's no one data model that works for everything. I think people are starting to gel with OMOP, but then in terms of a large set of VCFs for germline variants, or maybe somatic variants, There's no good solutions. The GA for GH is working on that. Some of the other challenges are just kind of the basic data privacy, loss of fidelity, missing data, bias in your collection data. Also, once you start to work in real world data, various groups will reach out to you and say, hey, we own that standard, you owe us royalties. So be aware of that. But there's huge potential in combining real world data as a control arm or in a pragmatic trial or pre and post time windows around a clinical trial. And then I'm going to skip that because I think I'm out of time and just say thank you.
Awesome. Thank you, Brian.